Good afternoon or good day, everyone. Uh, I welcome everyone to the first, well, really the second of our ReproNim webinars, a monthly series that will be talking about reproducibility in neuroimaging and is sponsored by the ReproNim project. ReproNim itself is a NIBIB funded P41, a biomedical technology research center that is dedicated to reproducible computation in neuroimaging research. My name is David Kennedy. I like to say I drew this short straw in a meeting of people and ended up being the PI of this band of characters. But I'm of course proud to do that and representing a really fabulous group of people who work together on these reproducibility topics. But as the nominal leader of the group, it's really my job to help promote the overall mission of this project and help promote that to the rest of the world and our scientific colleagues. It came to my attention recently that very often when we as ReproNim are out presenting things, we often very quickly get deep into the details of the projects we do. We'll have webinars and demos and presentations on lots of different software tools that we're developing. But we don't often get the opportunity to take a step back and sort of look at the sort of high level vision of what we're doing. Let's not do that in sort of a public way. So with the near beginning of this ReproNim webinar series, it seemed like a good opportunity to take one of our sessions and really try to give the high level overview of the mission of the project, of the types of things we're trying to accomplish, so that then the rest of our presentations, both from our own staff and from our related colleagues, can fit into this bigger picture. I was having a little fun in our sort of 5-minute wait up to starting this presentation with my little public service announcement. If you've just joined, please be patient. We're going to start in a little bit. And as I was doing that, even though I was trying to say the same thing in the same way each time, I was not at all reproducible in my way of even being able to say that. So it, again, just reminds us, or it reminded me certainly, that reproducibility is a hard thing. And even when you're trying to be reproducible, there's challenges to doing that. And to some extent, the only way to have actually had my opening announcement be exactly reproducible would probably be to have recorded it and then just played that back on a loop. So again, as I was thinking about that in our warm up to this, it was just like, okay, what we need to be doing in our research endeavors really is sort of recording and use that recording as the ability to then at some point in time be able to replay, to reproduce the events that happened. So this analogy of recording what we're doing, of monitoring the provenance, monitoring the exact details of exactly how things were done, is really key to sort of our overview, our overarching approach to reproducibility. So it just dawned on me as I was trying to do that, that that might be an interesting analogy. There we go. And I don't want to spend too much time on this slide, but again, as I prepared this presentation, and as many of us have been doing these types of presentations for a long time, I realized there's a lot of repro-isms that we like to use. And not that I'm condoning drinking games per se, but if one were to consume their favorite beverage, coffee, tea, or whatever, every time these various repro-isms came up someplace, that might be a fun game. Clearly ideas such as open by design, which we've stolen from Yarek Helchenko and the DataLad team and the NeuroDebian team, and sort of repurpose that to re-executable by design is a reproism that will pervade a lot of the things that we talk about. I and our statistical folks will often complain that every study we do ultimately is underpowered. We certainly, will promote the idea that everything that we do in the context of the neuroimaging experiment matters. So everything matters is one of my favorite reproisms. As we have gotten into this, much more this idea that efficiency is really key, that reproducibility without efficiency is not going to work. So really this mindset of efficiency is the new reproducibility will pervade things. Another reproism that will appear through mine and other talks will be, if it's not fair, it doesn't exist. We'll talk about fair in more detail, but the F of fair is findable. And so if something can't be found, it might as well not exist. And then ultimately, we're really working on social change and sort of really changing the way we publish things. The current publication is really not serving us, the neuroimaging community, or the patient community, or the scientific community all that well. So all these things are sort of fair game. And if you have your favorite beverage nearby, anytime these ReproNims come up, I encourage you to enjoy that beverage. For the sake of today's presentation, I'll be following, this overview as here. We'll talk about the reproducibility problem. We'll get into my favorite topic of everything matters. We will introduce the revised publication concept of the re-executable publication that we refer to as the ReproPub. Faced with the challenges of trying to do reproducible publications, we will look at how to break that problem down into more manageable pieces, our so-called 5-step program towards more reproducible imaging. And then ultimately the meat of what ReproNim itself creates is the set of tools and features and best practices and things that make accomplishing these various steps towards more reproducible imaging possible. And so that's the ReproStack set of tools. We'll look at a more fleshed out example of a ReproPub, and then we'll wind up today's presentation, and hopefully this will set up things for future webinars. My particular background in the old days was the MRI physics, and I'm old enough to have been involved with a lot of the early developments of structural functional diffusion imaging back in the 1980s and 1990s and things like that. Being in the early days of those techniques, that was great. The Wild West was we could apply our new imaging techniques and our new image analysis techniques to almost anything that had never been done before, and that's great. So we could almost grab any 20 kids or 20 adults or 20 people, do some imaging on them, do some analysis, and publish that. It was novel, it was new, it had never been done before, and hope that we would find in short order with our new functional tools or our new structural tools or our new diffusion tools the keys to the various neurological disorders that we were turning these instruments to. Well, it turns out that while we were able to do that and publish all sorts of things and find all sorts of findings, much of this didn't really replicate or doesn't really generalize to necessarily the groups that we're looking at. So over my history, I've evolved from developing tools themselves to worrying about how to pool data together to get to bigger data sets. And then I've learned that even big data sets don't solve all of our problems if we don't use those data sets in a re-executable, reusable way in order to build upon our observations amongst each other. So I've kind of gone the gamut from tools to data sharing to this work that we're leading here now on the reproducibility. But we've been doing this, we, the community, have been doing this for a very long time. Neuroimaging, again, sort of started, to be very prominent in the 1980s-ish and forward. So one would think, one would hope that by now we've learned some useful things. One of the important topics, though, is that particularly in our thornier biological problems, and I think particularly a lot about psychiatric applications, we really haven't succeeded at solving too much from the patient's point of view. And I use as an illustration of this concept a quote from Tom Insel and colleagues that was lamenting that after 30 years of the National Institutes of Mental Health funding billions of dollars of imaging research, billions of dollars of genetics research, the kid who shows up in their child psychiatrist office with autism, the adolescent who has a alcohol addiction problem, the adult who has schizophrenia when they show up in their psychiatrist's office, the prognosis, the outcome, the interventions today are quite similar to what they were before this whole neuroimaging and genetics enterprise started. So to the end patient, we haven't yet really accomplished all that much that is useful to them. The flip side of that is we, or the research community, have done a great job. We've gotten lots of grant money. We've published lots of papers, and that's great. But that's great for us. It's not great for the for the patient. And so that was really sort of highlighted in a bit of words here that I liked from Tom Insel's group that really talks about the profusion of statistically significant but minimally differentiating biological findings, i.e. lots of papers, but that these really only have approximate replications when you try to replicate things. And these replications that are attempted are done in ways that neither confirm nor refute those original findings. So again, these examples of things that we're finding are not turning into things that are relevant to the patient, to the clinician, to the outcome of the situation. And then that's a problem. And eventually, that's a problem we'd better solve if we want to sort of stay working in this particular type of area. Now, there's lots of reasons for these types of problems, and this really was a launching off point from the National Institutes of Mental Health's point of view to remember that probably the things we label as diagnoses are not necessarily the things that are most salient in terms of the structural or imaging or genetic or types of things that we're looking at. Labels are often, many psychiatric issues are sort of spectrum disorders, are groupings of types of different types of patients together under a common label, schizophrenia, autism, but that those labels cover a broad set of different types of features that can go into the getting of that label. So that kind of spurred the assessments and the labeling of patients from the research point of view to not necessarily concentrate on the diagnostic label, but to look more at specific characteristics labels, the RDocs, the research domains of characterization that can try to probe to the actual features that do care more about brain circuitry, that do care more about specific genetic factors. And while they may not be specific to schizophrenia or to autism, they can be specific to anxiety or to socialization factors or to memory features that span across different diagnostic classes. So that was one type of shift that may help to try to get towards a basis that we can find the imaging or the genetic bases for these types of features in a way that can be useful in terms of diagnosis or monitoring effects of therapy. The other piece of the large part of variance and the types of things that limit the success in the past is the sort of way we do our neuroimaging publications. There's lots of variations, lots of things happen, and we don't necessarily report them completely. We report them in a nice word way, but we don't necessarily report them in a way that can really support the confirmation or refutation of the claims that are emerging in our literature. And we end up just building up a literature full of these claims that can never really resolve themselves. And so those are the types of background concerns that we're sort of trying to work on to do our sort of imaging side of trying to help move the field forward and see if we can actually make a more relevant neuroimaging approach to these different psychiatric applications. There's lots of reasons, as I was mentioning. Here's again another one of my repro-isms: is everything is underpowered, and we need to look for the right targets, and diagnosis itself may not be the right target, and we don't really publish things completely in our typical publication, and we tend to only publish the positive findings and thus build up a positive biased literature that doesn't self-correct very well. And again, not to belabor the point, most people who are listening to this probably mostly believe that there is a reproducibility problem. Although the interesting thing that I've heard, I might not be able to quite defend, but you ask a bunch of people, if there is a reproducibility problem, or if other people's research is reproducible. Often people will say, no, other people's research isn't reproducible. But you ask them if their research is reproducible, and they'll say, of course, my research is reproducible. So that's kind of a odd juxtaposition that everyone themselves thinks they're doing a pretty good job, but most people think that the field itself has an issue. So there has to be some sort of disconnect there that makes that possible. So that's just kind of an amusing thing. So anyway, the reproducibility problem is not just a genetics thing or not just a neuroimaging thing. It pervades lots of science, and this is just one little example from the more basic science, more drug trial, literature from Bayer Healthcare. Bayer was trying to develop cancer, take leads from the published literature, from the basic science published literature, and use those as leads to develop future pharmaceutical interventions. And that's a very hard, very expensive, task, and vast majority of those, clinical trials fail. So that led these folks from Bayer Healthcare to go back and ask themselves, okay, these leads that they're starting from, the basic science, publications that are implicating the various compounds that they're trying, well, how stable is that literature itself? So they went back to a set of publications and found that 65% of the leads of the publications that were generating leads that one would follow up on were not consistent, were not reproducible themselves, whereas only 11-ish% of that literature that was generating their leads was able to either be reproduced or mostly reproducible. So that's clearly evidence of a problem, that if it's the basic science that's generating the leads that the pharmaceutical companies then follow up and try to bring to a product, and more than half of those are problematic, well, we're seeding the drug discovery pipeline with bad data, and hence that's going to be bad for the outcome of drug development and drug delivery. And when that's bad for the patients who critically need these outcomes to work to alleviate their situation. So that's just another example that reproducibility is a problem. Again, not to belabor that, but just as a nice example to have in the back of the mind. So moving back into sort of an example of reproducibility in the context of neuroimaging, I like to use this little example. A paper I might be writing might have in its abstract the following statement: we see an increase in resting state connectivity between the right superior temporal gyrus and the right superior frontal gyrus in subjects with autism, and this connectivity is correlated with diagnostic severity. Fine, lovely statement. Most of us listening to that statement, roughly know, what that means. And we publish that, and you could discover that in your PubMed search, and you could cite me when you see a similar type of thing or a dissimilar type of thing, and we could anchor our observation relative to these kinds of sentences. But if you actually wanted to replicate the statement I just made at sort of the high level, abstract level of my article, you really have to dig much deeper into this sentence. Every word, almost every word in that sentence is nuanced by lots of things that add variability to how one would assess the reproducibility of that statement. So I'm claiming increase. Okay, well, what was my statistic, and what covariates did I use to make that assessment of increase? Resting state connectivity, fine. What kind of data did I acquire for that? What was the TR, TE? What kind of analysis did I do to make an observation about resting state connectivity? Between right superior temporal gyrus and right superior frontal gyrus. Okay, those are anatomic regions. There is, many different ways to define those regions, and exactly how well those are defined or not depends on how they're being defined and what kind of assumptions are going into that anatomic summary. So that's quite variable. I'm doing this in subject with autism, great, but what are the subject characteristics? How old are they? What's the gender makeup? What's the socioeconomic status, genetics, environment, et cetera, et cetera? That can all be quite variable in any group of a patient population. And this connectivity correlated with diagnostic severity. Okay, what measure was being used, and the like. Now, all of these statements, all these details are probably provided somewhere in the publication, and that's great. But if you really wanted to replicate this particular finding, you really would have to determine all those different features and to re-execute that particular question to see if you could replicate that particular finding. And so that's quite challenging, that to be able to do that in a precise framework. But that's just how hard it is to, and so it's not surprising that often when people try to go and replicate something, they don't do something exactly the way my original statement was generated. And so, hey, you may not find the similarity, the finding that I did, Is that because it isn't true, or is it because of some variance that you made in your interpretation of my statement? Don't know. We don't really control for that. We tend, as human readable, human analog creatures, tend to just try to work at that sort of high level construct without really being able to deeply dig into the electronic machine elements that pervaded that statement in the first place. So that helps us at ReproNim to frame our concept of reproducibility around this concept of re-execution. So again, when I write my original paper, I get some particular data, I choose a particular analysis, and I generate some particular result from which I make whatever my claims are. That's kind of like taking a flag and sticking it in the ground and saying, I found this particular result with this particular analysis and this particular data. The first level of things that we argue you need to be able to do in order to even think about the reproducibility of that result is to be able to re-execute that result. Can someone, yourself or in a future time or someone else, access that exact same data? Can someone, yourself or some other person, access that exact same analysis? And then when you put those two things together, do you get that exact same result? We'll argue that for the most part, and again, most things that I complain about the community is not doing are things that I've not done for the past 20 years as well. So I'm picking on myself as well as everyone else. But so fine, I probably can't go back to a paper 5 years ago and find that exact same data, find that exact same analysis, run it on the exact same computer, and generate that exact same result. And I'm without the ability to do that, it becomes very hard to generalize how well that finding, that result holds up to variations. And so that gets us to this point of really wanting to be able to scientifically, to precisely examine what we call the generalization space around that result. We need to be able to re-execute that result so that we can systematically look at if I take that exact same data and just make small changes or do changes to the analysis, how sensitive is my result to the exact details of the analysis? The exact tool I ran, the exact parameters I ran, holding the data constant, we should be able to explore the generalization space around similar analyses and see how sensitive that finding is to those kinds of details. A good finding should be relatively stable with respect to different tools and different parameter settings that are looking at that same outcome variable. Similarly, you can flip that around, hold your analysis exactly the same, and play around with the details of the data. If you used similar data, if you use more data, if you use less data, if you use other people's data of a similar population, does the result hold or not? How sensitive is your result to the details of the exact data set that you're putting into that analysis? Again, a good result should that is about the particular patient population that you're claiming had better be similar in similar data of that same class, or else there's an issue with the class, the way you're naming that class of subjects that you're using. So overall, a true result should hold up under similar data and similar analyses, and generate that similar type of result. Now, the problem with this slide, the way I have it here, is similar is a problematic word. Exactly how similar a result is when you change any of the data or any of the processing is still an interpretation by the end user. But we'll of course argue that should be an electronic interpretation, not so much a human interpretation. The example I like to give is two results. Think of a spot of functional activity that is sort of overlapping, but the centroids of activation are not in the exact same place. Well, if you are writing an article and really need to claim novelty, even though they don't exactly overlap, you can claim, my spot is different than the other people's, and that's great. I have a new spot and the other spot is wrong or something, if you want to claim the novelty of your finding. Conversely, if you're trying to build an argument for some other thing and you want to claim similarity, the fact that your two spots overlap a bit, you can claim, we found the same thing, these spots overlap, and then go off and use that for whatever purpose you need. So again, this similarity being a problem and how we tend to in the literature, use the concept of similarity in whatever way fits our stories is again another source of what is or isn't a reproducible or generalizable observation. Again, as already foreshadowed, many of the issues that affect reproducibility, low power, and again, this is back to my beverage consumption game. Everything is underpowered, especially in the psychiatric domains. I'm going to argue, and fine, argue with me if you like, but the types of things that we're looking for in the details of psychiatric symptomatology are exceptionally subtle. These are small effects, especially under the background that we know things that have much larger effects. We know that age, for example, has huge effects on how large a structure is, or how well connected it is, or how great its diffusion anisotropy is. We know that sex makes a huge difference on these things. We know that socioeconomic status matters. We know that genetic factors matter. All of these things have known large effects on most of our outcome variables that we measure with our imaging techniques. Yet we're looking for a second or third order effect, the effect that has to do with why I hear voices, or why I have little social anxiety in certain situations, or why this compulsion to do something. These are all markedly smaller types of effect compared in the presence of much larger effects of these other covariates. So ultimately, we're looking for very small features in the context of very large covariates that we need to handle. And so my general conjecture is almost everything we tend to do is underpowered. Even our large studies, our ABCDs, our human connectome projects, our ADNIs, ultimately are only barely powered if you want to control for all of the other factors that are important, that we know are already important. So if we can stop right here and think for a second, if my conjecture is true and everything is underpowered, we sort of have two choices. We could give up and not do neuroimaging research because it's just too underpowered and we're not going to find anything. And if that were the case, well, fine, we'd all stop listening to this presentation right now. On the other hand, my other conjecture is, just because things are underpowered, or we have low power in our experiments, doesn't mean don't do those experiments. But what it does mean is do those experiments in a way that they can be pooled and added and aggregated to the growing scientific literature about that particular topic. So the hope is that lots of low-powered studies can be done in such ways that they can be pooled and aggregated, and together make less low-powered studies as they get put together. So to me, the problem is not studies that are low-powered. The problem is studies that are low-powered that are done in a way that can't be pooled and completely described and fed into a bigger ecosystem of emerging studies that are based upon that. So don't do low-powered studies, but do low-powered studies in a way that fits into the giant ecosystem of observations. A lot of other details pervade the literature that we know about. There's mistakes, there's we don't share data very well, there's different ways to do the same thing. How we sample subjects and sample various things are a problem. P-hacking and p-phishing are a problem. And we've already talked a little bit about the publication bias as causing troubles in the literature in terms of trying to find out which claims are generalizable and what claims end up being reproducible. There are many solutions and many emerging solutions. And again, what ReproNim does a lot of work on is trying to improve the pile of solutions that are beginning to emerge to be available for people. This issue of low power, one of the solutions, of course, with that is, data publishing, data sharing. We used to call data sharing, now trying to take and give the author back to be in control of that, give more power to the collector of that data, the author of that data, and really make data publishing a common part of how you approach a study. Ultimately, what used to be one publication, your data and your analysis and your results, where the results are kind of the main thing and end up being a little bit fragile, really take that one publication and turn it into multiple publications. A publication about the data and that data, how it was acquired and how it can be reused in other contexts. A publication about the analysis or reuse of someone else's analysis could come out of a current publication. And then the publication about, sure, your conjecture, your observed results, which can have its own life. So what used to be one publication really could be 2 or 3 publications. Great for the author, great for your CV, and great for the community of being able to independently confirm, refute, make live or grow the data or the interpretations in a much more sort of structured fashion. Methodological variants, again, people just from the words of a paper don't necessarily do the same thing. So getting workflows specified precisely and standard execution environments are a tool. P-hacking problem I'm referring to, one of the solutions for that has to do with pre-registration of analyses. That's one of the tools. Publication biases, yes, your words may talk about a particular story, but we also have ways to distribute your entire set of results so that your negative results and the positive results that you didn't happen to talk about in your publication can still live on and be usable and feed into meta-analysis and things like that. And ultimately, and again, picking on myself, many of us have not really been as properly indoctrinated in statistical training and in reproducibility centric training. And so we really have to keep the nose to the grindstone and keeping our sort of statistical abilities up to snuff in sort of very tricky statistical types of things that we're trying to disentangle. So if we talk about re-executability, we remind ourselves that we're working in a very complex space. So there's data flows through many stages of analysis and operations on that. And again, each of these sort of become a publication opportunity. So instead of just taking our end publication, there's a lot of these features along the way that should be pertinent for documenting the research object, the thing that you as a researcher have done and should get credit for and should be able to independently derive and get credit for and share and reuse in meaningful ways. We start from the left side, the acquisition itself, and there's a particular set of subjects, a particular set of interventions you do to them, and a particular set of data that comes out of that operation. Well, one, that's a publication opportunity, and two is really characterizing that precisely is important, that when you go to share that or try to pool that with other things, we need to know exactly what was acquired. Next, you're faced with the analysis, and here begins to be some of the issues that with your one data set that you acquired, you are faced with myriad possibilities of how to approach the analysis of that data. Do you use SPM or FSL? Do you use FreeSurfer or ANS? Do you use these sets of parameters or those sets of parameters? And ultimately, you, the scientist, make an informed choice about a particular way to do that analysis. You do that analysis, and we need to publish and document precisely what that analysis was that you did, so that when you come to looking at the result, we know exactly how that result was derived. And again, that brings us to that result itself. We need to know how that was derived, but we also need to, as part of the publication, keep that entire set of derived data available. Just because I see a spot here, also by implication means I didn't see a spot in all these other places. And so that null literature needs to be as relevant as the positive literature. So again, this is a publication option in addition to whatever lovely words you want to talk about this spot that you saw. We also need to capture the entire set of data so that additional reasoning about people who do or don't see activations in other locations or don't see this particular activation can pool those together in a more appropriate way. But there's many different ways to accomplish these analyses. There's many different ways to interpret these derived analyses, and so we just have to be clear about writing down exactly which of those we're doing, so that when we explore other ways of doing that analysis or other ways of telling the story about the results that you saw, that's not inconsistent. We don't lose the ability to do all of that just because you picked a particular analysis pathway and picked a particular story to tell about a particular set of the observations. We just have to be transparent about all those things, not to not do the things we're doing, but to do the things we're doing in a way that opens up their transparency, opens up their extensibility to other reuses. Back to our beverage consumption game, everything matters. From the very get-go, the very ground of our processing, the operating system of your computational analysis matters. If you're running a analysis on Linux versus OS X system, you may get different answers. Simple little FSL-based volumetric segmentation pipeline, can give you different answers, on different operating systems. The differences aren't necessarily huge, and I'm not going to claim that this is the biggest problem of our reproducibility spectrum of things, but there are differences that exist. And in small structures, these small little voxel differences, can end up making, 4%-ish, coefficient of variation in small structures like the nucleus accumbens. And while that's, a small number of voxels, that is a moderate, fraction of that structure's variance. And if we're looking for small biological variances, that 4% anatomic variability may be in the same order of magnitude as the structural variability that we're looking at, for that particular, behavioral or biological change. So again, sometimes, these types of very small differences can be exactly in the same range as the types of things that we're looking for. Tool selection matters. Many publications around that remind us that different tools make different assumptions and come up with different types of results, even for nominally similar types of observations. This particular slide on the right talks about two different tools, FreeSurfer and ANTS, looks at a particular structure, the average thickness of the left caudal anterior cingulate. Start with this little panel down here, where FSL version 5.1 and FSL version 5.3, I'm sorry, FreeSurfer 5.1 and FreeSurfer 5.3, correspond, very nicely together, and that's kind of what you expect, feel like, two different tools, measuring the same thing ought to do. But if you jump to a different tool, in this case ANTS, the exact, relationship between an ANTS interpretation of the left caudal anterior cingulate average thickness and the FreeSurfer version of the left caudal anterior cingulate thickness can be less correlated than you think. So therefore, if you go off and make conclusions about such results, it may depend on exactly which tool you're using, and it's not the result, it's not the left thickness of the left caudal anterior cingulate that's the major factor. It may be something to do with the tools that are generating that. Now again, I'm not saying that either one is right or wrong, but just that there's differences, and so we just can't use the anatomic concept as the proxy for the fact that that's modulated by how we actually measured it. Number of data subjects in your data set matters. We know larger numbers of subjects in general generate larger power. A little simple example of this comes from, in this case, looking at an estimate of the sex difference in the hippocampal volume using sort of the available public data in 2005, the NIH pediatric data set, which showed a non-significant sex difference, but a pretty large error bar across those 325 subjects. Jump forward to 2011, when you add the PING, the Pediatric Imaging Neurocognition and Genetics Study, adding another 1,200 subjects to that, we see a now significant and much tighter variance on this measure of sexual difference in hippocampal volume. Jump further forward when you can add the ABIDE data set, typicals of the ABIDE data set to that. Now again, our N is larger, and that result is holding relatively stable. So again, just because something doesn't exist in our low-powered studies doesn't mean it doesn't exist, but it means it's something that should continue to be evaluated at larger and larger sample sizes, especially depending on our understanding of what the variance of these measures are and what the effect sizes that we're looking for are. So as I was foreshadowing, the ReproNim mission is really to push this concept of the ReproPub. The ReproPub is the re-executable publication. We define a ReproPub, as having all of our wonderful words as usual, that's fine, but plus some supplemental way of indicating the exact data, the exact workflow specification, and the exact execution environment specification, as well as the complete results that generated as part of the publication itself, so that in principle an independent person could do that complete re-execution of that project, of that publication, and that complete description requires the provenance of this data, workflow, execution, environment, and results. And again, if we publish this way, then our ability as a community, ourselves or others from the community, to be able to systematically explore the generalization of the finding and to pool this data or this approach with other approaches becomes much more natural part of the research endeavor. So often when I say, hey, we should make all of these things available as part of our publication, most people look at me and say, hey, that's kind of hard. If you're saying, we have to do all this other stuff, just to write our publications, it might take you a year to do something that you could otherwise do in a month. And on the one hand, that would be bad. That would slow people's progress down. That wouldn't necessarily be good. So part of our mission at ReproNim is also to try to be practical and realize that going whole hog, making your next publication completely re-executable is a really hard problem. But if we break the problem down into pieces, and if each of us in our next publication could pick one of these pieces that's accessible to us to do in a practical way, our next publication will be a little more re-executable, better designed than our prior one. And each time we do new things, if we can find these elements of these types of procedures that we can be a little bit better and a little bit better on, we don't, well, it'd be great to jump to the full re-executability from the get-go. The practical thing is just to improve practices as we can. And again, people will adopt better practices if they're more efficient, or if it's clear why the benefit that you get from doing things a little bit different than you do today. And so again, by breaking the problem down into these different steps, we can help people choose the pieces of this that are available to them, and we as a project also see the places where we need to improve the efficiency so that we can lower the barrier to adoption of these various things, particularly the ones that are difficult to accomplish. So we break this down into, at the point of study design, there are things we should really be doing a little bit different going forward. We really need to be building the shareability of data right into the consent form to start with. To me, it's unethical to use federal or funding money to acquire data that doesn't have some sort of shareability built into the acquisition process. We too often hear people saying, oh, I would share that, but the consent isn't right. Well, fine, you might have been able to get away with that a few years ago, but it's just, in my opinion, unethical to acquire data that isn't designed to be shareable from the get-go. So we just have to get over that and insist on getting that built into, or at least the option for that built into our data acquisitions. Pre-registration is important, and again, to be able to plan the study out appropriately ahead of time is very critical. And part of that planning of the study out is to be able to know ahead of time the types of data that are being acquired and the semantics and the markup of that. It's much easier to mark up exactly what you did at the time you're planning the study, as opposed to 5 years later when someone's insisting that you share the study. 5 years later, it's hard to remember exactly what you did. When you're designing the study, though, that's when you know exactly what you're doing. You know exactly what behavioral measures you're doing. You're knowing exactly what clinical measures you're taking. Well, mark that up then, and then you'll never have to worry about it again, and you can go along with your sharing and your publishing options with less service later. On the data collection side of things, we need to be using standards as as possible. BIDS is, one of the important brain imaging data structure for managing imaging data. So doing that and using tools like ReproNim to get your data into BIDS is a nice way to go. Doing the markup of your metadata is important at this data collection point in time. Collecting that data in a provenance-aware type of environment is clearly important. And another big issue is, many of the software developers, out there, are very comfortable with version control and software, and that's great. The whole concept of version control as data migrates through the, ecosystem of an experiment is critical as well. So the idea that data, should be, as amenable to version control as, software and code, and that environments, are as amenable to version control, so that, you can monitor the provenance of what you're doing and be able to roll back or go forward or document, where you end up in the garden path of designing your analysis. And tools like DataLad, of course, are useful for that kind of thing. In the processing of data analysis, we need to take control of our operating system, our environments, which are a source of variance that we tend to ignore at the moment. So doing things under containers of some sort, Docker, Singularity, et cetera, and being able to do those in sort of version controlled ways are critical. And then marking up the results, not just your free surfer is free surfer, but being able to mark up the nature of the results, volumetric results, thickness results, et cetera, so that when those do get pooled, the provenance of the tool that you used and the provenance of the type of data that it is declared and monitored, so that it's easier to pool that information with other things later. Statistics, what really can I say other than, it's hard, and many of us are not, as well trained in all these details as we should, but we often, are a little bit, I'll use the word naive exactly about how we use p-values and confidence limits and significance testing. So we really need to pay attention to that more closely and, stay trained and up to date on sort of the best ways of approaching those. And publish everything. So issues related to that, there's no reason that what used to be one publication should not be 3 or 4. And things that we publish, things that we do need to be findable, need to be accessible, need to be interoperable, and need to be reusable. They need to be fair. So ReproNim itself, therefore, is, in addition to some of the existing tools in the community, are working on tools that facilitate various elements of the steps that we would like people to take better control of for themselves. There are search and discovery tools. How do we find publications? How do we find data from the various data repositories and publication repositories? How do we mark up our data from the get-go with proper semantics? How do we manage our input data, our DICOM data, and get control of our data from the get-go? How do we manage our computational and execution environments in ways that are efficient and yet also, especially as we get into bigger data problems, scale nicely across the different environments that you may be faced with dealing with, from your local computer to your local high performance computing to your potential to use cloud computing. As part of the marking up of our data, we need to be using common schemas, and so we need to take care of those schemas. As we use containers, that's great, but we need to be able to share those containers and distribute those containers in an efficient and accurate way so that someone else can get the container that you're using and have a chance of doing the type of thing that you're doing. So we need support for that kind of functionality. The slightly amusing, at least to me, aside is we've been suggested to work on our branding, and so of course you see Repro in front of almost everything. The one I could not get myself to put Repro in front of is this concept called TestKraken. And it's a tool that does generalized regression testing of scientific workflows. So like we would regression test software, we should be able to take that same concept and regression test our workflows, regression test our results against parameter settings and operating system settings and various things. But the TestKraken logo was just too good that I couldn't undo that. So unfortunately, doesn't have the repro kraken in front of it. Anyway, so all this is designed to help us generate, help the community generate more re-executable, fully provenance-aware publications. And then there's a large training component, both from the ReproNim training core and inserting training functionalities, like the train track aspect of the OHBM brain hack. So given that our time is really pretty much up, the shared slides will give you an example of a sort of fully re-executable publication. The one I alluded to earlier was a simple, super simple, doesn't really do anything biologically, but was a paper that was designed that you could, there was a quick enough and easy enough problem that you could actually re-execute that whole publication yourself, just to prove that these technologies work. In reality, we're working with larger data sets and things where you don't really want to re-execute someone's paper, but you do want to know that you could re-execute someone's paper, and you do want to know that you could generalize someone's paper. So again, the next version, so that first publication, the simple one publication, was one that sort of just proves the concept, and now simple two, which is still a work in progress, but has a abstract back from the OHBM meeting, is looking at a much more interesting biological question, looking at the literature and a bunch of hypotheses about structural correlates of IQ in typically developing children. And so can we take our known public data resources that have children imaging data and have IQ, and can we use the data ohm of the world to generate a repro-pub that looks at the generalization or the reproducibility of some common conclusions from the literature. And so in this case, there's a bunch of papers, 3 different papers here that have specific hypotheses about what we should see with respect to brain imaging and IQ. So all these turned into sort of 9 hypotheses, are pre-registered, right? There's a bunch of workflow stuff that we have to handle that all needs to be sort of done in a sort of re-executable framework. We have a container that runs the particular analyses that we're going to do, FreeSurfer, FSL, and FIRST, and ANTS. This gets containerized using the NeuroDocker facilities, run out onto the Amazon Web EC2 of the Amazon Web Services through AWS Batch. Runs on, runs on these public data sets that have imaging and IQ available, and then the results are published as well. So we can look at our various hypotheses. This first one was that total brain volume will correlate positively with IQ in both sexes across the complete age range. So we observe a similar thing, and so we've confirmed that first of those in a now completely re-executable way, as opposed to the the meta-analysis that that original finding was from. So great, that could be then extended and can live as more data becomes available, and probed more explicitly and examined if different tools or different statistical approaches alter that main conclusion. So all in all, there's 9 different hypotheses that came from the literature. In our particular analysis here, the ones that are green here are our analyses that confirm some of the analyses from the literature, some of the hypotheses from the literature. The ones that are red here are where we don't confirm the hypotheses from the literature. So somewhat interesting examples is the MAC3 hypotheses said that the left striatum volume in under 20-year-old females was not significant, whereas in our little study, we see a significant correlation between left striatum volume and IQ. So maybe that one was a little bit underpowered, and with this better, larger data set, that one becomes accessible. And sort of conversely, there are places that showed a negative correlation with the corpus callosum, in children under 12 in both sexes, and we don't see that in our particular data set, etc. So this is just a way to, again, take some hypotheses, but the key here is that we're reproducing or testing the reproducibility of those hypotheses, but in this case doing it in a completely re-executable fashion that again sort of opens up the ability to do those more generally as you go forward. So I'm going to end here, just back to our overall mission at ReproNim is to push the tools and the efficiency of getting through these various broad categories of neuroimaging research. There are a lot of people who are involved with this, a large set of collaborators who are listed here and available at our website as well. And I'll conclude by saying that, beware that we are now starting this monthly seminar of ReproNim details based on this sort of introduction. We'll be probing into lots of different details, lots of different tools, lots of different tasks over the next months. And so mark your calendar now for Friday, December 6th. We will be taking control of your data from the DICOM, from the scanner, using ReproIn and HudaConf, and Yarek and his team will be presenting on that then. And so stay tuned to our website, our Slack, our Twitter, our email, etc., to stay in touch with us.